Hello, everyone. Can you hear me? Yay! Fantastic. So good to see everyone. Hello and welcome everyone to the second SCNAA Gathering pre-conference meeting. My name is Casey Craig and you know me as Stella's mama, but recently I've had the pleasure of volunteering for the Acute Syndrome Foundation by organizing the Family Book Club and moderating the Clinical Trial Readiness educational series that we all thoroughly enjoyed last month. We are 15 days away from the 6th annual SCNAA Clinician, Researcher, and Family Gathering sponsored by Neurocrine, Praxis, Zogenix, and Greenwich. I think we can all agree that we would rather gather and hug each other in person, but I'm sure that we're still excited because we can still enjoy this amazing event virtually. With that said, as we prepare for the upcoming gathering, the Acute Syndrome Foundation is hosting pre-conference meetings where we will discuss topics that's important to our families. Last week, we had a wonderful discussion with the ladies from the Danish Epilepsy Center. So if you missed it, definitely go back and watch that recording. Today's meeting, we have 4 amazing researchers from the Patel Lab who will be presenting their research at the gathering. The topics for today revolve around their SCNAA research, and they can give us maybe some more insight on how to better intervene and possibly a new approach for treatment for our kids. Parents, we are not researchers. Well, some of us aren't, right? So sometimes I know I can get lost in the detail of things. So today's discussion is meant to be a casual conversation for us to become more familiar with the science so we can better understand their presentations at the gathering. So we invite you to ask a lot of questions throughout the discussion today. Okay, before we get started, we do have a few housekeeping items. So group interaction through questions and or comments will take place in the chat box only. So the chat box is to your right and allows you to type questions or comments, responses for Hillary or our researchers. To your left, there's a tab titled Expo. And this area you will find troubleshooting if you're experiencing any type of technical issues, or you may also type a request in the help or for help in the chat box. And our fabulous technical guru, Megan Barner, will help you tackle any of those technical issues you might have today. I will now hand over the mic to Hillary Savoy. She is the director and founder of the Acute Syndrome Foundation, and I don't say this flippantly. She truly cares about each and every one of our kids and fights for them every minute of the day. And lately at night too, which I have proof of her Facebook message at 1:41 a.m. this morning to prove it. Without further ado, our amazing leader and friend, Hillary.
Can you guys hear me okay?
Yeah.
Okay, wonderful. Thank you, Casey. Casey never fails to embarrass me just a little bit before I get started here, which I think is healthy, keeps me on my toes. So thank you, Casey. I just want to, since this is the last time we'll be meeting ahead of the gathering, I just want to give a shout out to Casey for having done just an amazing job being our host and moderator. Every time I dropped a bomb on her that she had to do this again, she just said, sure, no problem, I'll be there. And same for Megan. Megan has done a huge amount of work in the background for these events and for the upcoming gathering. And literally everything that I ask her, she just does. And she's an amazing resource for our community. And she's always behind the screen instead of in front of it. So just if you guys get a chance to talk to her, please tell her thank you. So welcome everybody. So happy to be here again for our last time before the gathering. As Casey said, we're 15 days away. I'm really excited and also exhausted from all of the work to get ready. And we're really excited to see all of you there. So that will be our 6th annual clinician, researcher, and family gathering. And as Casey said, we're so sad not to be in person, but also excited by the possibility of, what we can offer to families who wouldn't have been able to travel anyway. So we're reaching new people. And one of the benefits and the challenges of our gathering has always been that we have a really diverse audience at the gathering itself. It's a benefit because we're bringing together families, researchers, clinicians, industry to have conversations to help us all establish common ground and share what we know. But it can also be a challenge, I think, for our families to feel intimidated by some of the information that's being shared. So we've always done pre-conference educational efforts. And this year, kind of on the heels of the CTR series, we thought we would carry over those casual conversations to help provide a venue for families to educate themselves ahead of the gathering and to stay connected. So as Casey said, today we're joined by the Patel Lab from the University of Virginia. Those of you who have attended previous gatherings are, of course, familiar with Dr. Manoj Patel, who is an electrophysiologist and holds our SCN8A community near and dear. He's been involved to help organize really every single one of our medical and scientific professional events. He is our go-to for SCN8A research. He is an amazing collaborator across all of our SCN8A research network. He has explained science to our team at TCSF whenever we ask, both via Zoom now, but certainly also in the bars on occasion. And he has a wonderful team working with him in the lab who are also always willing to engage around the science of SCN8A. And as we've said in previous meetings here, you know, these conversations, this is how I've learned what I know, such as it is, about the science. And we've really wanted to bring that to all of you guys. So without further ado, I'd like to welcome the Patel Lab to press your share audio and video. And we're going to be joined by Manoj Patel, Ian Wenker, Jeremy Thompson, and Eric Wenger. Hey, Jeremy.
Hey, how you doing?
Well, thanks. Hi, Ian.
Hey, everybody.
Glad you made it. This jump, for everyone who doesn't know, we're in Zoom together, and then we make this leap over here together. We never really know.
I was nervous.
Everyone has made it. Manoj is here. Eric's here. Wonderful. So the gentlemen from the Patel Lab here are going to be sharing with us a couple of papers. One called "The Tonic Phase Apnea is Required for SUDEP in a Model of SCN8A Epilepsy." And I guess, Jeremy, I didn't have a title for yours, so I made one up. Impaired astrocyte function contributes to seizures when we see in SCN8A epileptic encephalopathy. Did I do okay?
That works.
All right. And they're also here to help us sort of dig into the context of their work and how it fits into the broader SCN8A research landscape. Before we start with Ian's presentation, I just want to give the families a heads up. We are going to be talking about SUDEP, I think, as you can tell from one of the titles. And we are going to be watching some videos of mice experiencing seizures and death. This is part of the reality of scientific research. I know for some of us that's difficult, and for others of us it isn't. So just wanted to give you guys the heads up. If you are uncomfortable, need to step away, ask kids to leave the room before we get there, please do so. And as always, these panel discussions are very casual. So please feel free to ask questions in the chat, interrupt these gentlemen. They're used to hanging out with all of us. They know we're nosy and have questions, so don't be shy. I think they'd be disappointed if you don't interrupt them. And before we start with Ian with your full presentation, I'd just like to ask you guys to go around, maybe starting with Manoj, just introduce yourselves, give some background on your work and your relationship with SCN8A. Oh, you're muted, Manoj. It's right at the bottom. It's like the video and then the. No, okay. Well, while you're.
Jeremy, you want to go? I'll run. I'm not far from him, so I can go take
Okay. Okay. Cool. Yeah. So hello everyone. I'm Jeremy Thompson. So I joined Manoj's lab a few years ago. And that was my start in the field of SCN8A epileptic encephalopathy research. I kind of brought some knowledge with me studying these other class of cells in the brain called glia that I'm going to introduce today. Maybe some of y'all have heard of them. And so since September 2018, I have been doing research trying to understand how these cells impact the disease. And I know I met some of y'all last year in Baltimore, and that was really cool experience. But that's about the gist that I have for myself and studying. So I'll pass the torch to Eric.
Can you all hear me? Okay, great. So my name's Eric Wingert. I'm in, I guess, 5th year now, graduate student in Manoj's lab. So I've been there working on SCNA my whole time, and so I've actually had the privilege of meeting probably many of you. I will miss being together in person, like was already said. I've enjoyed the last 2 years doing the SCNA 101 talk, and I'm working on that again for this year, although we'll be online. I'm hoping, if you haven't seen that talk, I hope that will be another level of where space where you can ask questions and learn a little more. Yeah, so my research is mainly on looking at neuronal mechanisms of SCN8A, and so we're trying to identify which neurons are causing the disease. And so there are some neurons that I think have been considered not to be as important. Inhibitory neurons, that's actually what I'm working on right now, and I'm hoping to wrap up soon with my PhD and with that work. So hopefully that'll be available for you to look at soon. So thanks for having us.
All right, I can go next before Manoj. My name's Ian Wanker. I'm a postdoc in the lab. Been here for a little over 2 years. I think it was like 5 years ago we met on a plane from a neuroscience meeting, and he started describing the mice that you guys have. And I knew a little bit about SUDEP based on the research that I'd done, more basic research that I'd done previously. And since then, we formed, a friendship, I'd say. And it took a little while, but eventually we started doing these experiments, and that's what I'm going to talk about today. So it's been a good collaboration between the two of us.
Yeah. So sorry, looks like I'm having technical problems over there, but I'll go back in a minute so that Ian can take his mask off. Thanks everybody for joining. I can see some of the names coming up, and it's when I get to meet in person and drink a beer, after the session. It's one of the highlights of my year for sure. I love meeting all the parents, all the children that they're able to travel, and hearing your stories. It's really dear to our hearts, and we really are trying to do our experiments so that we can learn more about the condition that these children suffer with, and the hopes that we can have some therapy in the future. So I've got a wonderful team, and you've heard Eric before, and it's a shame he is going to be leaving us soon, but he's going to go onto a bigger and better lab and do even more wonderful things. So we're excited for that. And so hopefully you'll get a good idea of the kinds of research that we're doing to help better understand the disease. So again, don't be shy in asking questions. As I've always said, there's no such thing as a silly question. So please take advantage of the fact that we're here and we're happy to help you understand what we're doing and the disease itself. So with that, I'm going to head back to my office and let everybody else take over.
Thank you, guys. And as you can see, I think in the chat, everybody is a huge fan of the 101 session. Certainly, Eric. And I think, as Manoj said, having you guys join us for a beer after these sessions every year, I think is so important for our community. I think we joke about it a lot, but it's also really where we connect as humans and understand that we're all just humans trying to help these amazing kids. And I think it's really meaningful to the families to get to meet you in that way. And also families, I think maybe we underestimate what our interactions with researchers mean to them. And Manoj has said, he has all of his magnets up from every year from our poster with all of our kids' faces, and that really has been part of driving him forward. And I think it's important that we all understand that we're a part of this equation too, as family. So with that, Ian, do you want to talk to us a bit about what you're going to be presenting?
I would love to. Should I share the screen now, or?
However you want to proceed. If you want to just jump right in.
Yeah, I'll share it.
Okay.
Let me select the screen.
And a reminder to the families, if you want to make the screen with the slides bigger, just double click on it and that'll take over most of the screen.
And then I guess one thing I was going to say, I don't know if we mentioned this before when we were talking, if questions come up in the chat, if somebody wants to just yell at me, if I don't see it right away, then I can look at them.
Absolutely.
Yeah, so are we seeing we're seeing the page, my screen?
It's coming up. There we go. Yep.
But I don't have it in. Oh, I see it. Okay. One second. Sorry.
As I told Jeremy, the worst thing that happens here is you get teased terribly.
Yes.
So.
Not that one. Green. 3.
So we're seeing, I think, your maybe your desktop, like background picture.
Yeah.
Perfect.
Now there's a page. Yes, perfect. All right. All right. Sorry. Yeah. So I already introduced myself, but and I mentioned kind of how Manoj and I got together on this topic. In the past, I've done basic research in rats and mice, trying to understand how the brain controls our breathing and our cardiovascular function. I think originally I was drawn to these things because they're very tangible, real behaviors that we all have to deal with, and they can become extremely critical, of course, under certain conditions, such as during an epileptic seizure. And so I kind of renamed the title to what I sent Hillary yesterday. It's Mechanisms of Breathing Dysfunction During Seizures. I think is kind of a general way to look at a large fraction of what I've been doing. And so, like everybody said, please interrupt and ask any have about anything. I think I have like 11 slides, and if I go smoothly and everybody likes it, that's great. And if there are any questions about anything as I go along, please just type them in there, and I'd love to just talk. And I'd like to know what people think is interesting and what people think is confusing that I'm working on. So. I mean, a lot of stuff is freezing, like a lot of it.
There, yeah, you were out for a minute, but I think we can hear you now.
Now?
Yes, you're back. I think he disappeared again. It seems like maybe his connection isn't great. Can one of you guys maybe text him and have him refresh his screen, and then maybe Jeremy, we can talk about we can just jump right into yours.
Okay, yes. So where is oh, here it is. I think this is the screen share. Why can't, so I go to my screen share and share is kind of grayed out. What is it?
So you press the little monitor button,
Yes.
and it's grayed out for you.
It's grayed out for me. Share is grayed out for me.
Oh, there's Ian.
Yeah, he's back.
And Frozen. See, I shouldn't have said anything about not having technical difficulties this time. Casey was right. So Megan, do you want to pop on and see about helping Jeremy share? There she is.
Hi.
Hey. Yeah, I see your message. I found it. I'm going to the monitor, but yeah, it's just.
I wonder, do you have?
Oh, did you? I bet you it's. So do you see where it says your entire screen, application window, Chrome tab?
Mm-hmm.
I would go to application window and then click whichever.
That's it.
Corresponding to whatever you want, and then it should light up.
Okay. That is it. It's working. Okay, so.
Thanks, Megan.
Yeah. Okay, so I can go ahead while Ian is troubleshooting. Okay, so hello everyone. So as Hillary said, my project is looking at the role of astrocytes in SCN8A epileptic encephalopathy. So I'm going to do it in this presenter mode so I can still see you guys and see the chat. Actually, no, that might not work. Okay, so I think most people are familiar with neurons as a cell type that is in the brain, but there's also this whole other class of cells. And I have a little cartoon of this very small piece of brain showing we gotta have our two neurons here, but we have these other cells, and these other cells fall into this class called glia, and glia come in several different flavors. So we have these microglial cells. These are these little immune cells, and they're present all the time. They're in my brain right now. They're in your brain, regardless of whether or not there's an infection or an injury. So they're these little immune cells that hang out there. You also have these oligodendrocytes, and here's one in blue. These actually wrap neurons. And you might be most familiar with these cell types if you've heard of the disease multiple sclerosis. So it's these cells in that disease that preferentially are affected and die. But these cells, their main function, you can think of them as helping neurons communicate faster with one another. And then we get to astrocytes, which is the cell type that I'll be talking about. And these cells have a whole bunch of functions, but they're very, very diverse functions. And so I'm going to talk about a few of the functions that these guys have, just to kind of frame their importance to the brain overall. So over here, I have a section of mouse brain, and I've labeled the astrocytes in green. So you can see them, they're all throughout the brain. And some of the functions that they have that are really important is forming barriers. So they form the inner lining. You can think of it as an inner lining to the brain. And I'm going to show you that in a second. But also, they contribute to the formation of the blood-brain barrier, which is
Jeremy, you're muted.
time. Or did it?
No.
Oh, that's so weird.
No, just now.
Okay. So that's weird. All right. So I guess I'll start at the beginning of the slide.
You said the blood brain. You got to the blood brain barrier, so it was.
Okay. Awesome.
You were just about. Yeah.
Okay, that's weird. Okay, cool. Okay, so to orient you guys to these barriers, I have a human brain here, and we're going to focus on this little slice, and that brings us here. And you can see here's the skull. We have the scalp up here, and then you have the outer covering, the meninges. And then to see where astrocytes contribute, we're going to zoom in even more to this little piece. And here we are. So the bottom half, the pink half, is the brain tissue itself, and we can see the astrocytes in there, and they have these little arms, and their arms go up to the surface of the brain and form this barrier. But they also send little arms to wrap the blood vessels. And so if you look over here in the image of the mouse brain, you can actually see those barriers. So
Jeremy, sorry to interrupt. I don't think that everyone can see your entire screen. I'm wondering if you can go to a different,
like this?
different view. Yeah, just because some reason, is everyone else getting it cut off? I think that there's an issue with that.
Okay,
Yeah.
then I'll keep it like this. Okay, I don't think my animations will work this way, but that is okay. Okay, so you can see these barriers in this image that I have. So these strips of green up here are actually the astrocytes putting their little foot processes at the end of the brain. And then you can see these two little lines of green right here are actually astrocytes wrapping a little mouse blood vessel in the brain. So this has a protective function. But they're also involved in neural communication. So we think of signals being sent in the brain from one neuron to the next, and involving exclusively neurons. And neurons talk to each other at these little sites where they touch, called synapses. And I've blown one up for you here. And neurons talk to one another by sending these little chemicals to one another, represented by these little triangles. But actually, astrocytes are present at these synapses too. And their main role is to kind of act like sponges. So they soak up this extra neurotransmitter. But they also soak up other things, principally potassium. And this is really important. I'm going to talk about that in the end of my talk, their role there. And so since they're so involved in protecting the brain and in ensuring neurons talk to one another appropriately, you can imagine that they may have a role in various diseases. And so these are publications by other researchers where they've discovered roles for astrocytes in various diseases, be it neurodevelopmental diseases, Alzheimer's disease, multiple sclerosis, but also epilepsy. And so in other forms of epilepsy, astrocytes have been studied extensively, but very little work has been done in SCN8A. So I bet a few of you are familiar with Miriam Meisler. She has some work looking at gene expression in astrocytes in this disease, but that's about it. And so my project is really looking at how are the functions of these astrocytes changed in SCN8A. And one of the hallmarks that astrocyte researchers look for when they're asking the question, are astrocytes involved in the disease process that I'm studying, is their shape. And astrocytes will change their shape when they're experiencing an insult to the brain. And here I've labeled astrocytes. So I can take a brain slice and I can stain it so that I can visualize the astrocytes and see them under a microscope. And I'm using this astrocyte marker. It's not really important what it is, but we call it by this acronym GFAP. And in a healthy brain, you can see the astrocyte is kind of small, but in the diseased brain, this green signal, or my astrocyte marker, is way brighter, and the cell, it looks kind of swollen, and its arms are much thicker. And people who study astrocytes like to use the terms reactive to describe this cell type. This is important not only because it indicates to us that astrocytes are involved, but it also tells us a little bit about how their functions might change. So we know that when we see these changes in shape, that they correlate to changes in function. So, and changes in the normal function of astrocytes. So what happens when these astrocytes change shape is you get a decrease in the machinery needed for their normal functions. And you also get an increase in that marker that I showed you, which is why the cell looks so much brighter. So I looked at some of the mice that we have that model SCN8A, and what I saw was in a healthy control mouse, very few of the astrocytes express my astrocyte marker, GFAP. But when the mice are seizing, a lot of these astrocytes start to express it, and it gets really bright in a whole bunch of these cells. And we can quantify that over here to show that we do have astrocytes that are reacting to these seizures and that are changing, presumably changing their functions, which is what this shape tells us. And one of the functions that I was very much interested in is their control over potassium. And the reason I was interested in that is because neurons need to have the potassium controlled in order to function properly. And I want to demonstrate to that for you here is whenever, here's our synapse again, and whenever a neuron signals to another neuron, it spits out potassium into the space around it. And astrocytes, here we have 3 astrocytes, they need to be there to act as a sponge to take up this potassium. If they don't, too much potassium outside of the neurons will cause them to become hyperexcitable, which is one of the hallmarks of seizures in epilepsy, is to have hyperexcitable neurons. And so this is a very important function of astrocytes directly linked to epilepsy and neuronal excitability. And so I wanted to look at that in the astrocytes I see in our mice.
Pause.
Sure.
Looks like we have a question about, so if you go to the slices from the mouse brain, that's what it is, right? It's a mouse brain?
Yes.
So there's a question about if it's the same mouse at a different time. I'm assuming it's not the same mouse at a different time because it's the brain of said mouse. So
oh, yes. Okay, I get it. Yeah, so back here,
yeah.
Is this the same mouse or is it 2 different mice? It's 2 different mice. So the healthy control mouse is a mouse that has what we call a wild type or a non-mutant so NAV 1.6 sodium channel. And then the seizing mouse was it has the mutation in the sodium channel. And then we video monitor them to make sure that they were seizing. So it is 2 different mice. Yes.
So the mice are, because we have another question here, to reduce mouse variability. They actually are fundamentally different because one is genetically typical and the other is genetically has an SCN8A mutation. But otherwise the background genetics should be the same on the mouse. Is that correct?
Exactly. Yes. So they're and they're also the same age. So the rest of the genetic background is the same, and they're age matched.
And the seizing mouse, presumably you see the mouse seize, and you sacrifice the mouse at that point.
Yes.
So families, just for that clarity, it's a bit tough sometimes for some people to talk about who aren't used to this, but yeah, the mouse, they wait until the mouse is seizing, and then at that point they will what they call sacrifice the mouse.
Yes.
Thanks.
Cool. Yeah, no problem. Okay, so yes, so this potassium regulation by astrocytes is critical to neuronal functioning. And this is why I wanted to home in on this first to look at that. And what I see, so this is a lot to walk through here, but the key point is that we're able to monitor how much potassium is flowing across the astrocyte. So we're able to monitor how much potassium the astrocyte is taking in. And we can do that we have a little pipette, a little glass pipette that we can actually attach to the astrocyte. And we can monitor the potassium flowing across the cell. And what I see, these are the current traces of potassium. What's important here, though, is to see that in the control mouse, who has the typical sodium channel, we have a certain size of current, potassium current. Whoops. But in our seizing mouse, you can appreciate that this current is smaller. And so what this smaller current tells us is that in the seizing mouse, the astrocytes are taking up less potassium. And you can see that in a different way down here as well. In blue, we have the healthy mouse, and in orange, we have the seizing mouse. You can see that this line, the orange line, is below the blue line, indicating that there's less potassium being taken up by the astrocyte in the seizing mice. And so this could contribute to the seizure phenotype that we see in these mice. And so I have a lot more work to do to look at these cells, and other things could be other functions of astrocytes could be infected in this disease, but that is for ongoing continuing research. And so I think I'm about ready to pass the torch on to Ian, but I would just like to thank the members of the Patel Lab. You've all met, except Pravin, who is phenomenal and does a lot of our mouse surgeries for us. So, and with that, I'm going to stop sharing so I can see the chat, and I'm open to questions.
So I think one of the big questions that, and we'll see, I'm not sure if you guys know if Ian is he, oh, there he is. Hey, do you want to, Eric was able to answer one of the questions in the chat. Gail has another question. Do astrocytes have directionality like neurons? Gail's awesome. Just, I see the impressed looks on your faces.
Yeah. That's actually a really good question. So during development, they definitely have some directionality. So they're involved in the development of the brain. They'll extend a process from the top to the bottom. That directionality or polarity is kind of lost in adulthood, but there is an argument to be made. I showed that they were attaching to blood vessels. And so there is an argument that there's some polarity in that regard, where they're juxtaposed to the blood vessel versus juxtaposed to the brain tissue itself.
Okay. And Gail has a follow-up question. White blood cell count increases during seizures. Is the increase in astrocytes also an indicator of brain injury and inflammatory response?
Okay, yeah, so that's another great question. So going back, I'm actually gonna go back to share my screen so I can get to that slide. Yes, this one right here. Cool. So yeah, when I showed this data initially to Manoj, the question he had was, do we have more astrocytes in this brain? I can label astrocytes using a different method, not relying on the GFAP marker that I've talked about, and I've counted them, and I don't have an overall increase in astrocytes. So even though it looks like there's more, they're just increasing their expression of that marker to indicate that they're reactive. But to the rest of the question, in different scenarios, an increase in astrocytes could be an indicator of brain injury. So they're involved in scar formation, like if you think of spinal cord injury, they come in and proliferate at the site of injury, but not in this case. And in those cases, it could be a sign of an inflammatory response as well.
And Gail also asks, as reported in the literature, NaV1.6 expression on excitatory neurons is predominantly during development. Does this pattern occur with NaV1.6 expression on astrocytes? And could this implicate increased risk for seizures during spikes in development?
Yes. So far, there's no literature showing that there's a developmental increase in NaV1.6 on astrocytes. I have tried to detect NaV1.6 on astrocytes using the same methods that I've done for the astrocyte marker I've shown. But I do have an experiment that we're going to do in collaboration with Miriam's group, actually, where I'm going to send her, I'm going to isolate some astrocytes and send them to her, and she's going to look at gene expression to see if they express this channel at all, because that's one thing that we don't know, is whether or not these astrocytes express this channel at all. There is some work from another group showing that when these astrocytes become reactive, that they will actually increase the expression of this channel. But so far, I have not been able to detect that.
And I think Chenzia's question gets to what I wanted to ask you as well, which is, understanding that astrocytes play a role. Would this be suggestive in understanding with that none of you are clinicians, and so this is asking you to extend scientifically a little bit. Would it be suggestive of a different standard of treatment if astrocytes are playing a role in epilepsy in 8A?
Yes, that's kind of the thing that I always say is, astrocytes are important because we can open up these new avenues of therapy potentially. Right now, we're not at that level. There is some work so that the way that astrocytes soak up this potassium, they have a potassium channel that does this, and there's been work demonstrating that different anti-epileptic drugs will cause the increase of this channel in astrocytes, and so that could be one way to target astrocytes as a therapeutic. Right now, there are no small molecule drugs that we can use to increase the expression of this channel as of yet, but it is something that is very tantalizing. It's very interesting to think, like, if they are deficient in their potassium regulation, let's try to find a way to increase the expression of this channel in astrocytes, or any of the other molecules that they may decrease their expression of when they become reactive. So it certainly is, it opens new therapeutic avenues for sure to understand what exactly is wrong about these astrocytes in this disease.
Wonderful. Thank you so much.
Mm-hmm.
So with that, I think, Ian, do you want to give a whirl again and
try again? Hopefully it works a little better. I think you
we're
wouldn't
doing
think
science
the
here.
internet at UVA would be worse than at people's homes, but I guess from some trouble. So right now everybody's video is coming in really well, so that's a good sign, I think.
Okay. Ian, just in case if it goes down, do you want to send your stuff over to me, or
I can do that? Yeah.
I hate to do that, but or I could just go to Buster and buy you some time, but I just let me know how I can help, I guess.
I like that we're demonstrating scientific method here, that we're going to just try different things until something works.
Well, that's pretty much it.
Yeah.
What's going on here?
Ian, I could also show just like the very beginning of the other paper. I know that's different than what you have, but might be enough to just get the gist.
Which paper? Sorry, if you want to go ahead, I'm gonna talk.
It's a bummer. You're gonna miss out on Ian's excellent style.
And we were able to answer the NAV 1.6 question, I think, it looks like. Thank you guys for handling that.
Now would also be a good time to, I don't want to move in a different direction, but to buy Ian some time. Sometimes I know it can be intimidating with scientific talks. So I wanted to invite any questions that were, if someone is sitting there thinking, I really feel like this is a dumb question. If you are having that feeling right now, I would invite you to ask that, because someone else may also have the question.
Yeah, and those are really the best questions. Okay, so we have a question. How much correlation is there between the oxygen saturation level and the interruption in breathing during a seizure? The reason for asking is that sometimes when seizing, he seems to desat really abruptly. What other factors are in play to affect this data point?
That sounds really good. So
that's an Ian question.
Your oxygen, yeah, that's a good one. I don't need slides. So oxygen saturation in your blood is, oxygen has an input and an output, right? You're breathing and also blood flow for the input, but also activity. And seizures tend to do two things simultaneously, or at least more serious convulsive seizures do. That not only do sometimes, as I'm going to show you, people stop breathing, but also, there's a large amount of activity going on. At a regular and resting an individual, your brain is consuming, the estimates range a lot, but 20 to 30% of your metabolic demand. So during a seizure, your brain is much, and then you're having convulsions where you're, contracting muscles like you're lifting weights throughout your whole body or something like that. So there's a huge metabolic demand. That's probably why the oxygen drops very quickly. You could also have your heart slow, and I think that could cause larger nadirs in the oxygen saturation. But I think that the reason is twofold: that they may not be breathing adequately, and just the metabolic activity that's going on. I don't know if you got the talk, Eric.
Did you email it? It could be taking some time.
I did. It's taking time.
Do you mind if
it does look like Mary has a? Oh.
You go ahead, Hillary.
So Mary is asking, I guess this is a question for Jeremy, would the potassium absorption be different for loss of function versus gain of function in SCN8A?
Yes. Yeah, I saw that. I'm ask her a question for clarity. I guess, yeah, so she means loss of or gain of function for the SCN8A?
Yeah.
I could speculate. There are definitely mice that have a loss of function or decreased function of the SNA mutation. No one's looked at astrocytes in those mice, but I would speculate that if the astrocytes do become reactive in those mice, which there is a possibility that they could, I would expect to see the same trend. So typically, when astrocytes become reactive, you get the downregulation of these things. It's very context dependent, so there's no definitive answer until you look. But I would think it would go with the trend that you would still see decreased potassium absorption by astrocytes in those mice, or in that condition.
That's interesting, because that would, I guess, it would suggest therapeutically, and again, this is conjecture, but that one of the big discussion points among our families at this point in identifying loss of function versus gain of function is the difference in treatment avenues for those individuals. And so if the therapeutic approach was to increase potassium in both a loss of function and gain of function, in theory, they would be potentially receiving the same treatment and responding.
Yes, but I think also that even though the astrocytes might be reducing their potassium absorption in both cases, it's whether or not you even want to address the astrocyte potassium absorption in a loss of function setting. Maybe in that setting, it's not contributing to the pathology you see.
So Minaj has said, in my understanding, loss of function does not yield seizures, so it's possible that astrocytes are not altered, which is really interesting. And I think this is a perfect example of the conversation, scientific conversations versus clinical conversations, exactly. Because the loss of function mice do not yield seizures, but the loss of function people do. And they're different. And I think that's, it's such important for the families to know this isn't a direct correlation. Eric, what?
Oh, sorry, I was going to add the caveat. Mm-hmm. I mean, I'm not sure if we have identified people who have a single loss of function mutation yielding seizures, but we know that biallelic, so if you have actually two, so the Danish group, along with our help, published a study that showed that if you somehow get two loss of function mutations in SCN8A, then you can have epileptic encephalopathy. But it's complicated to us. We don't quite yet understand exactly how loss of function, gain of function, but I think most of the variants that we've analyzed are gain of function.
Yeah, and there definitely are presumed loss of function non-dual allele individuals who have epilepsy, they typically have less severe,
Okay.
Significantly less severe epilepsy, and it's typically later onset.
Okay, great.
And that's a super interesting portion of investigation at this point, because it has been entirely understudied due to that sort of presumption at a scientific level with the mice, and the way that they were expressing their phenotype.
Yeah.
And again, for the families, this is so important to see, right, that there is this difference. Mice are not humans.
For sure. And one thing I like to add, and I think I'll speak only for myself on this, because I think it's a little bit, I don't know if all researchers would agree, but it seems to me that SCN8A is kind of like sitting in a Goldilocks zone of not too much this and not too much that. So I think we seem to be where you can have it needs to be a very precise balance, and loss of function could lead to epilepsy, but also gain of function could do so as well. So it's very fine-tuned system, and that's what we're dealing with. And maybe mice are slightly less sensitive, like than humans.
Absolutely.
Ian, how's it going?
I think Ian may have frozen again. So I think what we can do, a couple of things. Ian, if you can hear me now, maybe we could have him record his talk, so that we can absolutely distribute that to the families and anything that he wants to say. And because I don't want him to be stressing for the last 8 minutes about coming in and out. And maybe Eric and Jeremy, I'm going to guess that you guys know some of his research enough to talk a little bit to it. And
yes, can I do that, Jeremy? Is that okay?
Sure. Yeah.
I'm gonna give a unprepared talk. The good news is I actually have some data I can show, so I'm gonna try to do that. But I think before even showing, I want to give you a sense of what his, where he's coming from and what he's oriented to try to understand, verbally without showing anything. And so you can ask questions, so feel free to interrupt. I'm going to try to keep an eye on the chat. So Ian he came to our lab. He spoke a little bit about that. He's really a amazing in vivo electrophysiologist, or a physiologist, and so he's able to do things that are quite challenging. So one of what Ian has decided to try to look at is the question of why do these mice sometimes have seizure-induced death? So why do they actually have fatal seizures? I see Ian's joining in. Ian, I'm giving you a prep if you can hear me.
I can hear you.
Sometimes they have seizure-induced death, but it's unclear why. And so Ian has actually developed in our lab a way of looking at some of the other systems that might be failing in response to seizures. So, for example, when the mice or when humans die, it's usually the seizure is disrupting basic functions, either in cardiac function, so in the heart, or in breathing. These are we consider vital physiology. And so Ian is really looking at how does seizure impact these in order to understand and hopefully try to not just understand, but to then prevent the SUDEP aspect of SCN8A. And so what Ian is able to do in the lab is simultaneously record EEG, so function of the brain, that can detect seizures. He video monitors them, so he gets to see how they move and behave. He also has them in special chambers he built to measure their breathing. That's called plethysmography. So he'll be able to tell if they're breathing or how fast they're breathing, if they have disruptions in breathing. And then lastly, he hooks up a couple additional recordings to be able to get EKG or electrocardiogram to measure the heart rate. And what this allows him to do is to examine when mice have seizures and die, which of these systems is failing and what's the order. does the heart stop right at the beginning of seizure? Then you'd think that maybe this is a cardiac failure, cardiac arrest. Does breathing stop, or is it some kind of combination? And he's able to examine spontaneous seizures in this way. Ian, are you able to present? Are you there?
You're nailing it. It's not like when I try to connect the screen. I tried disconnecting my computer from like all the other stuff that I have going on, and it doesn't seem to help. Manoj is also trying to bring it up.
I think something weird must be going on with the internet in the lab because
I couldn't even send you the email.
Yeah.
With the I've heard what Eric said, and he's dead on with what I would show are some things that I've shown before. If you've been to previous meetings, when we're able to record these mice, and I think monitored SUDEPs in patients, there's about like 11 people, because it doesn't happen that often, fortunately. And we can't have any control over that system. But in mice, we can breed thousands of them, and I can record them. And our mice don't receive treatment. They usually have to live by themselves to do the measurements that I do. And so we can observe these events in a controlled environment and actually try to understand what's happening to breathing and heart rate. And I think what Eric talked about already was that the main observation that we have initially is that breathing stops during a seizure, and while the heart continues for sometimes 10 to 20 minutes. And if the breathing doesn't recover immediately after a seizure and it stays, they stay apneic, then they die from that seizure. It doesn't always happen. Many seizures are very similar and they recover. And so one goal of future research is what determines these events in the brain. And so that's one thing I would show. But they always have this apnea during a tonic phase. And so that was the other part of what I was going to talk about, is they have tonic phase apnea. And the tonic phase is when you have axial muscles contract tonically for, up to 30 seconds or so. And this causes apnea from what we can see. And so one of my hypotheses was that we actually tonically contract respiratory muscles because breathing is actually a motor behavior that requires muscles to contract and relax over and over again every time you breathe. And so if they contract tonically, it's basically like a breath hold. So you're forcing yourself to not breathe. And recordings that we've done recently where we show that these muscles do contract during the tonic phase. There's no way to selectively stop them from doing this, but we can observe that it does happen. And then propose some future experiments where I'm going to try to understand the neural mechanisms, because we think it's a seizure that's I think is spreading to the brain stem, the bottom part of the brain, that we don't talk about quite as much, particularly in epilepsy, which is outputting to your body and creating these behaviors that are, it's the behaviors that we're most concerned with, right, in a seizure, the motor patterns, the stopping of breathing, and these things. And so trying to understand the neural circuitry that causes this to happen could lead to therapeutic avenues for us to intervene and at the very least, take seizures and make them less severe, and take away the parts that are the most heinous. So that's in a short phrasing what I was going to show you today. I don't know if Manoj is having any more luck than I am at the moment trying to get it up and running.
Well, and I do
Ian,
think,
are you okay if I show some of this data from the other study that we worked on together? Or?
Yeah. Bring up.
I think he froze. But
whatever you have.
I just want to be careful about our time, because the families will probably be dropping off around 4. So what I'm wondering, if maybe what we could do, Ian, is have you record your talk that you were going to give, and then we can put that right up in the support group, and all the families can watch it, and we can put it side by side with this video where we will put it up online. And there was a question here from one of the parents about find all of your research to talk. So if we could compile, you guys maybe send me some of the links to the research that could be shared with neurologists. And JP, we can help. It looks like Chenzia is already talking to you about getting you in contact with a mother, a French mother, and we can help try to find a professional in France who's interested in SCNA. We're happy to continue chats with you about that. But if that, does that work well for you, Ian? We'll just make more work for you.
Yeah, I'll do that. That sounds great.
Any other?
Yeah.
Okay. Cami says low potassium in blood can cause irregular heart. Could this be the same in SUDEP?
So I've never heard of anyone looking at potassium levels in the blood during the, related to fatal seizures, or in epilepsy, although that might have been done, and I'm not aware of it in the clinic. My understanding of that would be that you have some difficulty with your kidneys if you have high potassium blood or something else like that. But repetitive seizures, maybe that could be that could occur. But I haven't heard of that being looked at. There is certainly I'm going to talk about breathing, if that's what I was going to talk about, but there's a whole field of SUDEP research that's focused on cardiac mechanisms. A lot of mutations, not as much with SCN8A, but with a lot of potassium channels and other molecules involved in epilepsy, are also involved in certain heart arrhythmias, like long QT syndrome and stuff like that. So there's definitely a potential for a cardiac arrest. I would say that in most mouse models we have, this isn't what we actually see. So not only in SCN8A, but in like Dravet mouse models and some other calcium channel models, it's really seems to be breathing, which to me makes a lot of sense because your heart can function by itself, but breathing needs the brain 100%. And if your brain is incapacitated from a seizure and it's severe enough, this could be that. But that is an area. And so in patients, in the mice, it's one mutation. We have this like homogeneous background through all the mice. But in humans, things are going to be much more complicated, and there could be many mechanisms that are happening.
So I'm going to do one more question, and then I think we should wrap it up for today. So Kayla asks, wanted to make a comment for Ian and say that McKinley has had seizures with apnea episodes, and he helped me understand more of what's going on. I wondered why sometimes it was just her frozen tonic and not breathing, but all other episodes, it would sound like I would hear a clicking noise, like she was trying to breathe after hearing him talk. I think it was her going into tonic-clonic, but not so much clonic, if that makes sense. Yeah.
So it's like at the transition between the two. You probably have some fluctuations in the muscle tone. It could also involve your laryngeal muscles and things like that. There's often a cry at the beginning or at the end of a tonic phase. Is how it's described to me. I actually haven't witnessed that happen. Probably because your different muscles are becoming activated. You're forcing air in and out of these airways, and you may have some closure due to other muscles that usually close around the throat. It could cause different types of sounds I can imagine happening. That would be my guess on it.
Thank you guys so much for joining us, and I appreciate you hanging through the technical difficulties. And I couldn't imagine a better group to have a bunch of technical difficulties happen with, because obviously Eric and Jeremy were happy to jump in and help. And so Ian, you and Minaj are.
Yeah, thanks, guys.
I just want to thank you guys again for joining us, and also just for your dedication to this community. And every time we have had questions for you, you have done your best to answer them, even when they're sort of outside of what you're working on. And I know Minaj is the same. He always looks for answers for us, and we're deeply appreciative of that. And Eric, we are absolutely, I guess we will let you go on to another lab, but we expect fully that you will continue to
They do
be here with us.
They do SCN8A, so I may not stay. Yeah, he's still there, so I hope to stay
on
around. If you'll have me.
I'll send a letter. I'll send a letter insisting. Well, we look forward to seeing you guys in 15 days. And Eric will be doing the 101, and all of these wonderful gentlemen will be joining us again for the research roundtable, and Manoj will be helping to facilitate all of our conversations. So thank you again. And families, we won't meet next week, but we will see you the week after. So thanks everyone for joining, and we look forward to seeing you soon. Thank you guys. Bye.
Thanks, guys.